This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 243, recorded on May 27th, 2021. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. Nice to see you, all of you.
You too. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. Pleasure to be here.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
You guys behaving down there in the South?
We're trying. We're desperately trying.
That's good to hear. Yeah, well, here it's trying to be spring, I think. Looks nice. Looking nice.
We've
outside.
gone to summer, unfortunately. It's
right
already 90 degrees.
Wow.
Right now it's 93 degrees outside, and it's partly cloudy. And the rest of the week, it's going to cool off below 90. So that's actually nice.
But Michael, you're indoors, so it doesn't matter, right?
No, I actually freeze to death in my office. That's why I came home to record this, is because my office was too cold. And I said, you'll hear my teeth chattering.
Thank you. We appreciate it. And if you appreciate what we do here on TWiM, consider supporting us, microbe.tv/contribute. We have two cool papers, a snippet and a paper for you today. The first one from Michael is going to make you hungry, right, Michael?
That's right.
And so it is literally cool. Literally cool.
It is. The title of the paper is The Effects of Peroxyacetic Acid Spray and Storage Temperature on the Microbiota and Sensory Properties of Vacuum-Packed Subprimal Cuts of Meat. And it's from Yang, Wang, Rychok, and Clausen. And they're at the Agriculture and Agri-Food Canada, Lacombe Research and Development Center in Lacombe, Alberta, Canada, and the Cattlemen's Association in Calgary, Canada. And as we are recording this on the eve of the annual kickoff weekend for summer barbecue season, I thought it would be interesting to discuss the findings of this paper published in June of 2021 in Applied Environmental Microbiology in the subsection entitled Food Microbiology, in order to illustrate the rigor with which our food supply is protected and how sophisticated our understanding of modern packaging and the storing of our food can actually be. So well done to the authors and to the folks who actually funded them to look at this.
Michael, I have to point out, Michael, and Elio, listen to this. The editor is from the University of Naples.
Wow.
Danilo Ercolini.
Good place for food.
It's a good place for food. And he probably demanded ribeyes be sent. So one thing you got to keep in the back of your mind, these folks used 147 ribeye steaks. So as you go to the store this weekend and look at the price of ribeyes, think about just the cost of the supplies associated with this paper. So briefly, what we're going to learn about is how an application of a low concentration, and low I mean 200 part per million, of a strong oxidizing disinfectant, this is this peroxyacetic acid, applied in a commercial setting. What that means is as the ribeyes are cut, they are on a conveyor belt and they go into this little chamber that then sprays peroxyacetic acid on them. And then the conveyor belt rolls around, the steak is flipped, and again, it's sprayed on. And the questions that they are asking is how this simple act of spraying this small amount of a disinfectant on a food can positively modulate the meat microbiota of, in this case, the ribeye, so that when it's then vacuum packed and stored, whether it be at a refrigerated temperature of 4 degrees or some of the other storage temperatures meats are typically stored at, from 4 degrees all the way down to -1 degree Celsius, we can then assess whether the vacuum is breached, whether it actually tastes any different. And their score is effectively, they're looking as if it was freshly cut off the bone at the processing plant. First,
Michael,
to all the
Michael, what is subprimal? I thought they were.
We're going to get to that. We're going to get to that. I got that all factored in. First, to the vegetarians.
You can't wait.
I can't wait. First, to the vegetarians. I apologize for this story, but are going to make the argument that if we're going to continue to harvest meat as food, we should not waste it as a consequence of microbial spoilage. And this is now Vincent's question. Secondly, to all the foodies, as background, and in the show notes, I put a picture of a cow highlighting where the 8 primal cuts of beef come from. And the 8 primal cuts span the gamut from chuck, which the McDonald's hamburgers are made from, to the rib, where obviously the ribeye comes from, to the loin, where we get the delicious filet mignon or tenderloin, the T-bone, as well as Vincent's probably favorite steak, the New York strip. And then, of course, there's these other areas. Subprimal, specifically to get to Vincent's question, and I had no idea what subprimal was. It's the cut of the loin that's specific. So in the loin, there's the tenderloin, there's the strip loin, there's the short loin. And these are typically packaged and then sent to your local grocer, who will then unpack the vacuumed meat and then slice it fresh for you at the market. So, bigger pieces are packed up, vacuum sealed. But in this case, they're actually packing the ribeye that is carved from the rib section, which falls between the shoulder, which is chuck, and the loin, and it spans from ribs 6 through 12. Now, there's not likely a consumer of beef products or a listener of this podcast that has not lived in fear of getting a package of ground beef, or for that matter, even apple juice from the supermarket, wondering if they have been contaminated with E. coli 0157H7 or a Shiga toxin producing strain of E. coli. And Shiga toxin when consumed by humans, will result in a hemorrhagic diarrhea that can be life-threatening under certain situations. And in fact, in 1994, the USDA, Food Safety Inspection Service, addressed this by declaring that Shiga toxin produced by E. coli was a food adulterant, and subsequently mandated that all federally inspected meat processing plants in the U.S. and those facilities that export to the U.S., like the Canadians or the Australians or the Brazilians, needed to implement what is termed a hazard analysis and critical control point systems-based preventative control plan. And initially, they started out by spraying lactic acid on things to drop the pH to inhibit microorganisms. But as time has moved on, we have gotten more sophisticated with the disinfectants that we have used in order to preserve the quality of the meat to as fresh as possible. And again, many animals carry Shiga toxin producing E. coli, but the cattle are regarded as the primary reservoir. And again, that's where we need to concern ourselves with, is the primary reservoir. And the hides of cattle carry a large microbial load, and the animal is brought in literally walking, and it leaves as a steak. And so in the process of dressing the animal, they, have to debulk it of the microbial contamination. and they are trying to understand the effect of E. coli. And E. coli, we know, is detected frequently from the colon of animals, but in the case of ruminants like cows, and cows have 4 stomachs, the majority of the E. coli is found in the rumen wall. And I put a paper in the show notes for you to effectively see how folks have worked that out. So when you consider how this animal is dressed for consumption, the microbes can be fugitively transferred to the primal and subprimal cuts of meat during the dressing out of the carcass. To better control against the contamination via, these fugitive emissions, whether you spill some of the gut onto the meat, or various physical and chemical decontaminating treatments have been mandated and incorporated into this HACCP prevention control plan for beef that is consumed in most of the developed world. Now, peroxyacetic acid is a substance that is a highly biocidal oxidizer that maintains its efficacy even in the presence of organic soils. So that's important. A lot of disinfectants don't work when and I learned this from one of my friends who speaks good Yiddish, when there's a lot of schmutz present. And schmutz is an operational term for dirt. And it can be protein, it can be debris, it's just stuff that's on it. So the peroxyacetic acid serves to remove surface contaminants, principally proteins, not only from the carcass of the animal, but it's also used to clean that endoscopic tubing that just performed your colonoscopy. Because think of where that tube is going when it looks to see if you got polyps. Its mechanism of action of how the peroxyacetic acid works is it denatures proteins, disrupts cell wall permeability, and oxidizes sulfhydryls and sulfur bonds and proteins and enzymes. In other words, it's a cellular nuke.
But what does it do to the flavor of my ribeye, Michael?
That is the point of the paper.
Yeah, I know it.
That's the point of the paper. So what's the story? So here they address the impact of spraying only 200 parts per million on the subprimal cut, namely the ribeye. They took 147 steaks. They put peroxyacetic acid on half of them approximately, and peroxyacetic acid on the other half of them. And they simply asked, they then vacuum packed the steak, and they took the steaks out at various time points. And then they asked what was going on with the dynamics of the microbiota associated with the meat, both by conventional microbiological methods, plating, and then by 16S ribosomal RNA gene amplicon sequencing, as well as what Michelle's getting to, sensory assessments. They have a panel of 5 individuals who literally sniffed the meat, and they were calibrated sniffers.
Before or after it was thrown on the grill?
Before.
Okay.
And again, everyone who has ever cooked chicken or beef or pork and got it out of the refrigerator, we all take a sniff. And you have to remember how meat principally spoils is the microbes are actually eating the flesh. And the lactic acid bacteria principally consume glucose. And when they consume glucose, they principally make lactic acid. They're homofermentative. That's the only waste product they make is lactic acid. So that lowers the pH to inhibit the enterics, like the ones that can make the Shiga toxin E. coli, as well as if the pH is too out of whack, it can actually inhibit the Clostridia, which putrefies meat products and protein. And if anyone who's ever used chopped meat broth to maintain their anaerobic cultures, and actually sniffed it, you know the putrefaction that can go on by Clostridia. It's pretty intensive of what the smell was. And so what they learned is, again, as I said, half and half. Half were sprayed, half were not. They then stored the stakes at 4, 2, and -1 for up to 34, 104, and 180 days. And they were sampled for sensory assessment, which is smell and general acceptability by the consumer. And there's a defined method that they use that's actually described quite nicely in their methods. And treatment with this disinfectant did not affect the initial pH of the meat, which is going to Michele's question again. What did it do to her steak? It's not going to make the meat taste funny or rubbery or mushy. And that pH of a good high quality ribeye is about 5.48. And the controls just a little bit more. It wasn't statistically significant. And overall, the mean for the overall mean pH for the polyacetic acid treated meat stored at 4 to -1 degree C was significantly lower. So the PAA is actually lowering the pH of the meat as its storage length increases. And that will likely become important because what we're going to learn from their extensive sequence analysis is it favors the growth of leuconostoc on the PAA treated meats. And the PAA treated meats were superior over the ones that weren't treated. So again, none of the packs lost vacuum to a blown pack. Why is that important? Because when enterics degrade protein and when the Clostridia degrade protein, they make both acid and gas. And so that can actually violate the vacuum of the vacuum pack seal. It will actually blow it out, literally, hence the term the blown pack. So they were looking at integrity of the vacuum. They also scored the samples for both odor and overall acceptability, which is, again, a method I was not familiar. So again, take a look at the method in order to see what they did. But since some of you won't have access to this, the packs were opened aseptically by cutting the pack below the seal with a sterile scalpel. Immediately after opening, the odor acceptability was assessed by presenting the meat to a 5-member panel with previous experience of beef. So they graded it on 1 to 5. 1 was strongly acceptable and 5 was strongly unacceptable. And so it's a 5-point Likert scale. And then the meat was allowed to bloom or come to room temperature for 1 hour, after which the accessibility was then scored again on the same scale. As expected, the scores for both odor and overall acceptability deteriorated as a function of time. And the variability from the samples increased with the storage time for all 3 temperatures, which is really a cool graph. It's presented on the y-axis as an organoleptic assessment. It's the first time I've ever seen that word on a y-axis, organoleptic assessment. And they offer that the development of the sensory properties over time, they just didn't have a big enough sample size in order to get it to fit to this model called the Barany-Roberts model. And then their next set of experiments was the 16S ribosomal RNA amplicon sequencing. And what they found is that treatment did not affect the initial numbers of aerobes, lactic acid bacteria, or the Enterobacteriaceae before storage. However, the the disinfectant application delayed the onset of spoilage by 7, 21, and 54 days, respectively, at 4 degrees, 2 degrees, and then -1 degree, respectively. So as expected, the microbiological analysis called out a number of things. There was no significant difference in the alpha diversity, which recalling that alpha diversity means mean species diversity in sites or habitats at the local scale. However, they also did principal component analysis, which was a different story. And here the amplicon sequence variants for samples from the 2 meat groups, the control and the disinfectant, or PAA, were primarily separated into different clusters. And when you look at the principal component analysis, you don't need to be a statistician to know the 2 groups are really different. What they found is that during storage, the microbiota were primarily dominated by Carniobacterium and Lactobacillus and Lactococcus on the control meat, but it was principally Leuconostoc, another lactic acid bacteria, on the PAA-treated meat. The bad actors, Serratia, Yersinia, Clostridia, were identified by linear discriminant effect analysis as biomarkers on the control meat, and Clostridium was found in high abundance in samples that had the highest spoilage rates. Their conclusion comes from one of the more interesting sections of the paper, which was the correlations between bacterial counts, sensory assessments, and pH. But bringing this all home to the consumer, PAA solutions, when applied at low concentrations under a commercial setting, positively modulates the meat microbiota. And what was remarkable, when you dig into the data, you will find that PAA did not have any bactericidal effects for beef subprimals, which is very important for the high-end restaurant market that ages beef. Because if you want to age your beef and have a dry age or a wet age steak at these high-end steakhouses, if the PAA was going to change that initial, if you will, terroir of the beef processing, you wouldn't get as quality a product. So it was really a remarkable story that PAA was able to differentially impact the members of the microbiota for the better. And from the perspective of the consumer, this then resulted in delayed onset of spoilage of these vacuum-packed beef subprimals like ribeye steaks, stored at all 3 temperatures that we often find in our supermarkets, whether it be 4, 2, or -1 degrees centigrade, so that the ribeye that you're going to offer up to your friends and families this Memorial Day weekend is all the safer for researchers like this. And one final thought I had is, what became of those delicious 147 ribeye steaks that passed the sniff test? If they have any PAA steaks left over, they would always be welcomed here, at least by this person on TWiM.
Michael, so the PAA does not negatively affect the odor, right?
No.
And did they report on taste? Did they cook them up and eat them?
No.
I don't think so.
Oh, I think this should have been rejected until they did that.
I'm sure there were some secret lab experiments done, Vincent.
Sorry, Michele, go ahead.
So it might occur to some listeners like, boy, they're going to an awful lot of really specific detail and using, state-of-the-art techniques to investigate this question. But I was struck that they estimate more than 1 million tons of beef are traded internationally each year. So this is big business. This is really important. And it's great to see our knowledge of microbiology applied. And I'll just point out, this is the second in our series. We did the microbiome of sourdough starter last episode.
But eating meat is not great for you, right?
Especially ribeyes. It's one of the more fatty cuts.
I mean, we introduce problems by raising animals for food. The swine and chicken industry, which provides a lot of protein, also gives rise to new influenza viruses.
Lots of them.
The methane that's released from the cattle farms is not good for the climate. So,
Now, I will say that milk is a good thing. I like milk
and cheese. Don't forget cheese, Vincent.
Yeah, cheese. I forgot the cheese. But that I'm trying to cut out the red meat because I think it's not a good thing. Well, I've been trying for years.
So one factoid for you, Vincent. For viruses, the dose range to inactivate viruses, specifically poliovirus, is between 1,500 and 2,250 parts per million when the polio is suspended in yeast extract. So viruses need more PAA to be inactivated. The reason for that is, remember, it's like PAA is like a molecular grenade to a virus. It has to blow up in the proximity of the virus to damage that icosahedral capsid of polio and then take out its nucleic acid.
We don't have a problem, but I just want to say that we humans bring problems on us, right?
Oh, yeah.
We raise these animals. It messes up the methane. It messes up it brings new viruses. It gives us colon cancer.
Antibiotic resistance.
Use of
antibiotics in animal feed.
And what, in Japan, you eat too much fish and you get stomach cancer. If you want to eat exotic meats like bats and pangolins and civets, you get other viruses. So I don't know. I think we should reassess, but it's never going to happen because as you say, it's a huge industry and people make a lot of money off of it, right?
But to your point, Vincent, I just saw an article in the New York Times where they were talking, there are people that are vegetarian, for example, or pescatarian, whatever. There's a new one, climatarian. They choose their foods based on the impact that it has on our climate. So, which nuts require more water to grow, to help people make choices.
Yeah, there. I mean, what is it? I think a single almond takes like a gallon of water in California to grow. It's crazy.
Well, that's what always troubles me about almond milk. Is not only do you have to grow the almond using a lot of water, but to make almond milk, you have to add water to it as well.
But there are lots of options now. Oat milk.
Oh, yes. Oat, soy. One of the first problems that the domestication of animals caused the human race is, of course, tuberculosis.
Well, we got measles virus from cows when we started domesticating cattle. I should say cattle, not cows, because cows are female. When we started domesticating cattle about 15,000 years ago, we got measles virus from them.
Yeah.
So I have switched to, Beyond Beef, whatever that is, Pat Brown's pseudo.
The one that we did the experiment in San Diego when we were with Elio. We all had a Beyond Beef burger.
It was good. It was really good.
Yeah.
So I cooked that. It tastes great. And, as I said, I have to be careful because my parents both died of colon cancer. And so I moderate my red meat intake. All right. Thank you, Michael. That is, I had lunch already and I'm hungry, man.
Yes.
Well, and so, Elio, what do you have for us?
I have an article from eLife magazine called Asymmetric Localization of the Cell Division Machinery During Bacillus subtilis Sporulation. What's that about? The authors, by the way, are Kanika Khanna, Javier Lopez Garrido, Joseph Sugi, Kit Poliano, and Elizabeth Villa, all from the University of California, San Diego, so I know several of them. Kit Poliano and Elizabeth Villa are quite a combo. Kit is one of the leading lights in the use of the microscope in the optical microscope for studying bacterial structure, and Elizabeth Villa is one of the leading lights in the use of cryotomography and its modifications. I'll explain that in time. So, what's this all about? From bacteria on up, all the way to the cells of our body, most cells divide by binary fission. That is, they start in the middle, the process starts in the mid cell. usually by constriction, and proceeds until two resultant daughter cells become separated. Most often, the two sister cells are identical in size and shape. I find it boring. Bacillus subtilis and other gram-positive spore formers do things differently. In spore formation, the conventional path, which is followed which happens when you grow vegetatively, is altered. They now the two cells that are formed, the division does not take place in the middle. The two cells are different. I find that exciting. Okay. Now, spore formation is an elaborate manner. I don't think, have we ever talked about it on TWiM? I don't believe
Not that
we
I
have.
remember.
Anyhow, let me say something about spore formation. It's a elaborate matter. It involves making a division septum at a precise spot, which is away from the center, near one of the poles. Don't ask how the cell figures out where the spot is, because nobody knows. But anyhow, it's a different division than that in vegetative growth. So the result is an asymmetric division resulting in a larger cell, which is known as the mother cell, and a smaller cell, which is called a forespore, which eventually becomes the spore. All this is masterminded by a series of regulatory genes that kick in at precise times. So the question is, are the two septa, the septum that's made in vegetative growth and the one that's made in spore formation, the same? If you guess that they're different, you'd be right. The events, following the events after the formation of the septum during sporulation are just as fascinating, because in time the mother cell engulfs the forespore in something that looks like phagocytosis, which is a process otherwise pretty much unknown in the prokaryotic world. So eventually the spore matures, it drops the "for" from the forespore term and is released when the mother cell lyses. So this is what they asked the labs of Elizabeth Villa and Kit Poliano. They and the graduate students involved who they actually did the work. So but let me before I do that, I have to tell you something else about all cell division in bacteria, most cell division in bacteria. It starts when a somewhat magic protein called FTSZ, or if you're British, FTSZ, polymerizes into a peripheral ring, the Z ring, at the division site. And this ring shrinks until the two cells divide. FTSZ is a tubulin homologue, and the first one that was found in bacteria, and it serves as a scaffold for about a dozen other proteins, which make a division machinery which is called the divisome. Okay, so we're talking about the divisome here. In B. subtilis, gram-positive bacillus spore former, one such protein is called FTSA, and it makes FTSZ stick to the membrane. Complicated stuff. Now, the question is, are the septa in vegetative and sporulating cell the same? And the answer is no, they're different. In the vegetative cell, the septum is about 4 times as thick as in the sporulation. So I'll call them vegetative septum and sporulation septum. And involved in making the sporulation septum is a sporulation-specific protein called SPO2E that localizes on the forespore side of the sporulation septum. In other words, it's not on both sides; it's just on the spore side. This FTSA filaments do something called treadmilling. That is, they start at the head end and get rid of material at the tail end. When they do this, they allow the synthesis of this main cell wall polymer, which is peptidoglycan. So far, so good. Mm-hmm. This is sort of an introduction to sporulation in 3 minutes. The work and the discuss reveals that these FTSAZ filaments are only present on the mother side of the sporulation spectrum, which may explain why the sporulation spectrum is thinner than the vegetative spectrum. So this is all regulated by SPO2E. So how do they do this work? They carried out, they used cryo-electron tomography, which I'll remember is a way of essentially doing a CAT scan of bacteria. It's a bit complicated. I won't go into how that works, but just believe me, you get essentially a CAT scan. However, in other words, you can see in 3 dimensions what's inside the bacteria. However, bacillus is too thick. Electron tomography does not work for thick objects. Only works for, in fact, E. coli is about as thick as it gets. So Villa and others figured out a way to do this, a way to get around this problem of the thickness of the object. They use something called ion beam milling. Milling means that they cut out some of the thickness of the cell by getting rid of material, just by sort of cutting it off.
So they do that physically, Elio, or is that with, do they do that computationally?
This is done with beams called focused ion beams.
I see.
So it's like a lathe.
They shoot these ion beams onto the bacteria. The bacteria, they get rid of some of the stuff.
Wow.
Okay. So what's left can be used for cryotomography because it is thinner. Okay, this is a very fancy technique. It requires quite a bit of equipment. Doing the cryo-focused ion beam milling is not trivial.
And can I just interject?
Sure.
The first author, Kanika Khanna, and I have exchanged email, and she describes when she first met Elizabeth and was considering doing a joint project for her thesis between the 2 labs. Elizabeth, among other things, said, just remember the word cryo has cry in it for a reason. It's painstaking work. Yeah, the presentation here is just beautiful.
You'll tell us more about Kanika later, won't you?
Later, yes.
Okay, fine. So using these techniques of cryo-focused ion beam milling and cryotomography, they could see a lot. So they could see both the vegetative and the sporulating septa, the 2 kinds of septa, and they had a great deal of knowledge to their understanding of these processes. A number of other labs are involved in this kind of work as well. So this is sort of an ongoing subject of study. So the main thing they found is that in vegetative cells, the FTSA filaments are located near the cell membrane. The FTSC ones are away from it. They found that interesting differences in the architecture of gram-negative bacteria, such as E. coli, and gram-positives like bacillus, which some of it is explainable by the fact that, as is known among microbiologists, bacillus has a much thicker cell wall made up of a lot more peptidoglycan than the gram-negatives, which have a thin cell wall. So why are the 2 septa different, the sporulation septum and the vegetative septum? The authors propose that this may be because it makes, I think I didn't say that in time the mother cell engulfs the 4 spore. Yes, I did say that. Okay, so maybe having a thinner septum on the spore side, on the 4 spore septum, makes it easier for the engulfment. So that is, the article has an impressive amount of data dealing with other details of the formation of both septa. I won't go into that because it would take too long. The article is well worth reading. It's a very good article. And let me quote what they say at the end of the paper. These are the authors speaking. They say, our data provide a significant advancement in the understanding of the organization of the divisome, its regulation, and its role in septal peptidoglycan synthesis in Bacillus providing, to the best of our knowledge, the first evidence for the asymmetric assembly of the divisome in bacteria. In other words, the divisome that is made during vegetative growth and the one that is made during sporulation are different, and that's news. And they depend on the SPO2E protein and so forth. So they end up by saying this will aid in the development of new antimicrobials targeting the cell division machinery in important pathogens. So it's a good paper.
It's so impressive where we've come with imaging. Yeah, many people listening probably, we would go from the 10x lens to the 40 and to the 100x lens, and that was like the increase in resolution. And now they're using these amazing technologies with lasers and computation.
That's right.
Stunning.
It is stunning, yeah.
So the first author, this was her thesis work. She said that she grew up in India, in the city of Delhi, and was good at math, but she really had no personal experience with science. But she was good at math and was able to attend the Indian Institutes of Technology, where she got both a bachelor's and a master's degree in biochemical engineering and biotechnology.
Is this in Bangalore or someplace?
In Delhi.
In Delhi.
Yeah, so she
There
took a lot of chemical.
Like that, yeah.
She took a lot of chemical engineering classes, but also basic biology classes. But there is a professor at University of Wisconsin-Madison, Asim Ansari, and he set up a scholarship program to bring Indian students to do research in U.S. institutions. And she was able to get one of those scholarships and spent the summer there and developed just a love of research. That was her first time really having the experience of lab research. And she also developed a lifelong relationship with Professor Ansari. She said he's been just an amazing mentor. She then applied to graduate school and went to UCSD and rotated through a number of different labs. But when she joined Kit and Joe Pagliano's lab, she said she just fell in love with microscopy. They were doing such elegant cell biological studies and also bringing in the genetics. She loved being in the microscopy room. And she also notes that the Pagliano lab members were the happiest lab members she encountered in all of her rotations. And then
She's a wonderful person, Kid.
And it was during that time that Joe brought Kanika into his office and said, "Listen, we've got this new colleague, Elizabeth, and she is doing some amazing cutting-edge research. She's one of the only people that has this technology. Would you like to have a chance to see things that nobody's ever been able to see?" Well, she jumped right in it, but was prepared that there is a cry in cryo. But she also was really fortunate to work with two postdocs in the lab. Javier Lopez Garrido helped with the design of the experiments right at the start and kind of vision for the project, helped her with construction of the mutants. And then Jose Sugla was, she called the data ninja master, who helped her work out the analysis of all the histogram intensity, where you can take the imaging data and then pull it into numbers. So she was really appreciative for that experience. She said an especially exciting day was when she was in the lab, in the microscope room, and she was noticing these regular dots in a particular cross-section. And she didn't know what they were. She sent this snapshot to Kit by email. Kit got excited. And so she ended up staying there for another 5 or 6 hours that night, going through section after section of all her old data, looking for more examples. And when she did find one, she could tell she was onto something, and she was just so excited. And she realized, I am the only one in the world that knows this right now, and I can't wait to share it. So she really had one of those incredibly exciting moments. So she has completed her thesis work after really contributing to a really impressive body of work. She's now a postdoc at Stanford and is also involved in a number of other outreach activities. So, for example, when she was a graduate student, she was president of the Association of Indian Graduate Students. She also is part of an organization, the Science and Research Opportunities in India organization, that connects Indian people here in the U.S. with professional opportunities back in India. She has written some blog posts for Small Things Considered and also the American Society for Microbiology. But she also says that something that's really helped her get through this strain of the pandemic and graduate school has been she has started to do some crocheting. And she has got funding from the Awesome Foundation to make crochet portraits of Indian women in the sciences. And I've linked to the show notes. She's got a really amazing large crochet portrait of Janika Amal, who was a botanist, and another one of Kamala Sohani, who was the first Indian woman to get a PhD in science. And I've linked to it. I really encourage our listeners to take a look. It's amazing art. She also has been, doing what she can to help the difficult challenges that her people are facing back in India. So she's volunteering with the India COVID SOS nonprofit organization that is at Stanford that is being led by Manu Prakash, who you all may have recall his amazing talk at the American Society for Microbiology, where he described his Foldscope project. Mm-hmm. So he has got a low-cost paper microscope that gives really impressive resolution, and their goal is to use this to make science and the excitement of discovery accessible to people of all backgrounds around the world. So they are now fundraising. They have vetted a number of organizations. So if you are interested in contributing to the cause in COVID and to help our colleagues and friends in India get through this pandemic, you can visit the show notes, and there is a place there. She points out that it's really been heartbreaking to go through this pandemic and the strain of worrying about her friends and family back home, but it's been helpful to try to do something positive. So she ends saying, the pandemic is not over till we vaccinate everybody. So there's a lot of work to be done. She also has some advice for junior colleagues, and that is to don't be afraid to venture into uncharted territories, get lost in the journey of curiosity. You'll find something new and eventually figure it out. And she also says, remember that any journey is a marathon, especially in science. So do take time for yourself and your hobbies and your loved ones. She also is very grateful that she took a graduate microbiology course that Joe Pagliano and Elio co-taught. And ever since, she's been hooked on both Small Things Considered, the blog, and also TWiM and TWiV. So Elio, she says you've been an inspiration, and she's so glad that she got to know you during her time at UCSD.
She's a lovely person, I gotta say. Yeah. It's reciprocal.
That's great.
Nice. I noticed in her letter, she said, she had this data, and she said, I immediately sent a snapshot to Kit. And if you know Kit, she usually doesn't respond very fast to emails, but she did to this one. And I have to say, so I went out to their lab a couple of years ago and did a TWiM with them. It was November 2015. And so the result was TWiM 115, lovely episode where Kit and Joe both spoke with me. We have video for that episode.
Cool.
And I had a hard time getting in touch with them by email. They didn't. And in fact, like the day before, I'm trying to confirm, I think it was there for some other meeting, trying to confirm that I'm going out there, and they didn't answer. And it was, so I understand. Well, but in the end, it all worked out. And it was great.
Besides doing this cutting edge research on cell biology of microbe cell division, Kit is also a dean. And I believe she and Joe, you can help me on this, Elio. I think, haven't they set up a fluorescence-based screening assay for drug discovery?
Absolutely. They have a whole thing.
Yeah.
That's right.
So.
Yeah, we talked a lot about that on the TWiM, and we went in, and there's some clips of the microscope room with someone. Yeah, it's great. It's a lot of fun. And then they took us to dinner at some seafood place. So that was cool. And they brought their daughter, who didn't really want to come with us. I remember that very distinctly. Do I have to go? But she came. Yeah, you have to go. We're going. You're coming. This is dinner. That's great. Thank you, Michele, for that. That's really cool.
My pleasure. And thank you, Kanika, for your very thoughtful sharing of your story and also the work that you're doing on behalf of the people of India. Our hearts go out to you and yours.
What's in store for Kanika? Do you know? Did she tell you?
Well, she's doing a postdoc now at Stanford and is with Michael Fischbach and studying host microbiome interactions in the gut.
Cool. All right. That'll do it for TWiM number 243. If you want to see the show notes, we're going to have all the links to things we talk about in the show notes. That's at microbe.tv/TWiM. And if you want to send a question or a comment, you can do that, twim@microbe.tv. You know, the emails have kind of died in the last year or so. I don't know, getting a lot of them over at TWiV, but very few here. So get with it, folks. Send us some email.
We have to give something away. Maybe if we got steaks, we could give away steaks. Ribeyes would do it.
Or sourdough starter.
Or sourdough starter, yeah.
If you really appreciate what we do, consider supporting us. Microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter's at Small Things Considered. Thank you, Elio.
My pleasure. Thank you.
Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.